So, good afternoon, students.
Good afternoon, sir.
Okay, so any questions on haptens? Any question, anything you want to discuss? Anything which is not clear? Okay, so is the concept of hapten clear?
Yes, sir, it's clear.
Okay, so who is going to explain haptens to me? So students, raise your hands. Okay, so I can see few hands now. Okay, so let me start with Yuvraj, and then I'll come to Yukta and then Sanjana. Okay, so yes, Yuvraj. Yes, what do you understand by the term hapten?
Haptens are kind of molecules which mainly combine with a carrier or a protein in order to produce antibodies.
एक बार फिर से बोलना.
Haptens are molecules which mainly carrier protein in order to produce antibodies which binds to the antigen.
Okay, so you are saying haptens are the proteins which need a carrier protein so that they can generate antibody, right? This is what you said?
Yes, sir.
Okay, and Yukta, how will you define it?
Yes, sir. Sir, so haptens are the molecules. They are also known as incomplete antigens because they are unable to produce immune response on their own. So they require to form a complex with carrier proteins, forming
Okay.
and after formation of that complex, they can generate immune response, and after that, they produce antibodies.
Okay. Okay, so now let me share this picture and Yukta, if you can, or anyone else can try to explain this picture to me. Okay, let me show it. So, who will explain this picture to me?
Sir, can you please make it in a slideshow?
Yeah, I will put it in slideshow, but who is going to explain this? This picture, I think, okay, Shakti, good. Uh, Let me put it on full screen and then you can explain this picture. Yes, Shakti, go ahead.
Yes, sir. So in the first one, we have one hapten and the second one is a protein. So they are different. We have induced them differently. So in case of hapten, as we know, they can't produce their own antibodies. So no antibody is produced. And in case of the protein, carrier protein, the antibody against its epitope is produced. In the second one, we have combined them, and then we can see that antibody against the protein is also formed, and the antibody against hapten is also formed. So in case of hapten carrier conjugate, we can see that we have the antibody which is formed against hapten also, and we have one antibody which is formed against the that protein also, and we can have antibodies which are in common also.
Okay, so I think, yes, students, I think now it's clear. So she has explained it in a very simple way, right? Which is actually required. So if you inject hapten, right? Hapten is incomplete antigen, right? So it is antigenic, but it is not immunogenic, right? So it will uh, be there in the body, but it will not elicit an immune response, and no antibodies will be produced against this hapten, right? Uh, Now you have some other protein which is called a carrier protein. It's a big protein, okay? And this protein has its own epitopes. And when you inject this protein, this carrier protein in a rabbit or any animal, antibodies will be produced against it because, of course, this protein is visible to the immune system. Okay, now in third case, we are making a conjugate, or we are coupling hapten and this carrier protein, okay, and we are injecting this hapten carrier protein complex in the animal, okay, and now it will be generating antibodies against the hapten as well. Okay, so antibody against hapten, right? So in the presence of a bigger molecule, uh, it will also lead to generation of antibodies. Okay, basically it will lead to generation or eliciting of the immune response. Okay, so I think the concept of hapten is clear to you now. Still, if you have any questions, you can ask. Okay, so I will now show you a video on haptens.
In this video, we'll talk about hapten. Now, hapten are small molecules with relatively simple chemical composition, which are not immunogenic. They cannot itself create an immune response, but they might create an immune response when they're attached or conjugated with a carrier. Point to be noted that the carrier might be immunogenic itself or might not be. Now we know anything that binds to the T cell receptor or the B cell receptor, which is membrane bound antibody, is known as an antigen. Now, antigens can bind to the T cell or B cell receptor, but not necessarily they would evoke an immune response. So, T cell receptor and B cell receptor binds to the specific epitope of an antigen, right? Now, on the other hand side, those antigens which can bind to the T cell receptor or T cell receptor and can evoke an immune response, those are known as immunogens. So that is how immunogens are differentiated from antigens. Now, haptens are not immunogens by themselves because haptens are too simple. You know, when the antigens are going to be presented to the T cell receptor, they have to be served on the platter on MHC class 2. Now, MHC class 2 has a peptide binding groove. that binds to 16 to 18 residues long amino acid. Now let's say the hapten like this, which is basically a benzene ring like structure, it's like too simple to be specifically bind to the groove of the MHC class 2 and presented as an antigen. So it hardly binds with specificity in the MHC class 2. Whereas peptides derived from several pathogens, which has a length of 16 to 18 residues, can easily bind to MHC class 2 peptide binding groove and can be presented to the T cell or B cell receptor. And if they are able to activate T cell, then they are going to elicit an immune response and they would be known as immunogens. But haptens alone cannot do that. So that is why haptens are so simple. They cannot activate T cells. They cannot activate T cells because they fail to bind to MHC and to be presented to the T cells. So that is why they are not immunogens by themselves. But hapten can't be an immunogen, or they can be. It turns out that hapten can be an immunogen when they're only conjugated with an adduct and form an adduct. And this experiment was done by Karl Landsteiner, where simply he injected a rabbit with a putative hapten. So this is a symbol of the hapten we are using right now, and saw that in the rabbit bloodstream there is no antibodies raised against this hapten. Now, then he injected the rabbit with a specific carrier, a relatively complex molecule, which is able to bind to the hapten, but he first only injected the carrier and saw there is antibodies raised against the carrier. And when he injected the rabbit with a combination of both hapten and the carrier, a totally different antibody is raised, which is against the adduct, the carrier and hapten adduct. So it turns out that antibodies or immune response is going to be generated only when hapten is conjugated and forming an adduct. A live example, let's just take a live example. So we know that contact dermatitis is formed by poison ivy, right? So when poison ivy is sort of uh, touching your skin, then an active molecule known as urushiol is going inside. Now, urushiol is nothing but a benzene derivative, so it is pretty simple in structure. What happens is when urushiol goes in, it's an alcoholic form which is getting oxidized inside the screen to form quinone derivative. And this quinone derivative is highly, highly reactive. Now, this quinone derivative interacts with the endogenous skin proteins and form a hapten adduct. This hapten adduct, this urushiol hapten adduct, could be recognized by specific Langerhans cells and dendritic cells sitting in the skin nearby, and they can be presented to the T cell. And this urushiol adduct, not urushiol itself, can elicit immune response. And this particular molecule is involved in delayed type 4 hypersensitivity reaction. If you haven't watched that video, the link is given in the description. So that is how we understand that hapten, when conjugated with proper carrier, can elicit immune response. I hope you enjoyed this video. If you like this video, give it a quick thumbs up.
So students, was it clear? Anything you want to ask from that? Any question or query on haptens?
So I have a question.
Yes, Manaswini.
So you had said that. So we know that they can't. They don't have immunogenicity, right? So shouldn't that technically make them even more dangerous because they can just attack our body without the immune system defending our body because it's not able to?
Yes, it can, right? They can be dangerous. I think if we take an example, okay, I'll give you an example of nicotine. Okay, I hope you all know where we find nicotine.
Yes, sir.
कहाँ होता है nicotine?
Cigarettes.
Yes, right. So when you are smoking, you are basically inhaling nicotine, right? So it goes into your lungs, and through your lungs, it gets into the bloodstream. Okay. So is nicotine good or bad? So, people get addicted to nicotine, right? So, and if you are addicted, then you again and again and again you want nicotine, right? So you will always keep smoking, okay? So, uh, scientists they thought that can we develop a vaccine against smoking, right? So what they did, they developed antibodies against nicotine, right? So when nicotine by itself gets into our bloodstream, it's a hapten. No one is able to. The immune system is not able to see it, right? And it can do whatever it want to do, right? So it is making us addicted. Okay, so scientists are working on this principle. So if you can inject uh, antibodies against nicotine, so suppose a person smokes, right? Nicotine gets into the bloodstream. Our immune system will not recognize it, but if we just inject antibodies, so those antibodies will go and bind to that nicotine and take it out from the system. Okay, so I think with this example, Manaswini, your question is answered.
Yes, sir, it is.
So yes, it can be problematic. Okay, because in that case, our immune system will not fight. Okay. Okay. Any other question? Okay, students. So if no question, then we can have your attendance, and after attendance, we will start with antibodies. Okay. So students, be ready for your attendance. Adish,
Present, sir.
Adish, you were not visible.
So am I not visible?
No.
So am I visible now?
Yes. Okay, Atoeba. Atoeba. Ayushi.
Present, sir.
Sam. Okay, Devanshi,
Present, sir.
Ishita,
Present, sir.
Janvi,
Present, sir.
Kshitij,
Present, sir.
Manaswani,
Present, sir.
Namrita,
Present, sir.
Nandini, Nehal,
Present, sir. Present, sir.
Rupal, okay, Sahiba,
Present, sir.
okay, Sanjana,
Present, sir.
Shakti,
Present, sir.
okay, Shivani,
Present, sir.
Tanvi,
Present, sir.
Tanvi,
Present, sir.
okay, Tridev,
Present, sir.
Vivek,
Present, sir.
Yukta,
Present, sir.
and Yuvraj.
Present, sir.
Okay, okay. So Atoiba is also present. Sam is also there. Okay. So students, good thing is now we know what is an antigen. Now we know what is hapten. Okay. So now we have a very good idea about hapten, about antigen, right? We also know what is an epitope, right? So these concepts are clear to us. Now the next thing we have always been talking about is antibody, right? अभी हैप्टेन के केस में भी हमने कहा कि हैप्टेन के अगेंस्ट एंटीबॉडी जनरेट नहीं हो रही, बट जब हमने उसका कैरियर कॉन्जुगेट बनाया देन देयर वाज एंटीबॉडी अगेंस्ट हैप्टेन, राइट? देयर वाज एंटीबॉडी अगेंस्ट द कैरियर प्रोटीन, राइट? तो अब हमें ये समझना है कि व्हाट इज एंटीबॉडी, ओके? तब जाके हमें एंटीजन और एंटीबॉडी का कॉन्सेप्ट क्लियर होगा, ओके? सो today I'm just thinking I will talk about the structure of antibody and how initially the structure of antibody was deduced, okay? उसके बाद नेक्स्ट क्लास से हम एंटीबॉडी का स्ट्रक्चर इन डिटेल वी विल स्टडी, ओके? एंड देन वी विल स्टडी द डिफरेंट टाइप्स ऑफ एंटीबॉडीज. सो आई एम श्योर यू नो, और अभी सुना भी होगा बहुत बार, हम बोलते हैं आईजीजी एंटीबॉडी, आईजीएम एंटीबॉडी, आईजीई एंटीबॉडी, राइट? सो दीज आर द डिफरेंट टाइप्स ऑफ एंटीबॉडी, राइट? और इन टाइप्स की भी कुछ सब टाइप्स हैं, राइट? सो वी विल अंडरस्टैंड द स्ट्रक्चर, द डिफरेंट टाइप्स and the function of these different types, right? जैसे for example body के अंदर IgG has some different role, IgA has some different role, IgE has some different role. So all the different types of antibodies have some different role, right? So we also need to understand what that particular antibody is doing, what role it is playing, right? Why that particular type is important. What happens if we do not have that type of antibody, right? So we will be talking about this, and I think this will take around 2-3 lectures just to explain antibodies, okay? uh, I will check if there is monoclonal antibody in your syllabus. So if it is there, then I will take one class on monoclonal antibody. Okay. But before we get on to that, we need to understand antibody. Okay. uh, अगर हम एंटीबॉडी वर्ड को ब्रेक करें, सो एंटी मतलब अगेंस्ट, बॉडी, सो अगेंस्ट अ बॉडी, राइट, अगेंस्ट अ फॉरेन बॉडी, right? So this, it is something which is against a particular body, right? So this is antibody. And students, I'm sure you know what is the structure, what is the shape of an antibody.
Y shape.
It's Y shape, right? So ये जो Y shape है, okay, हम इस Y shape को भी समझेंगे in detail. Okay, so now just let me share the slides. जस्ट होल्ड ऑन, ये थोड़ा सा टाइम ले रही है ओपन होने में. Okay, so students, in the meantime, if you have any questions, you can ask. Okay, students, so we will start with the structure of antibody. Okay, so you already know it's a Y-shaped molecule, right? But Y-shape में, this is how actually your antibody looks like, right? So this is a molecule of antibody, and you can see there is so many things here, right? So you can see it has an amino terminus and the carboxy terminus. Okay, it has 4 chains. You can see 1, 2, 3, 4, right? So 4 chains, and these chains you can see there are a lot of SSS, right? So these SS are your disulfide bonds. Okay, okay, let me keep highlighting them. Okay, so you can see here these S's, these are the disulfide bonds. Okay, and you can see because of these disulfide bonds, this chain is linked to this chain is linked to this chain is linked to this, right? So the disulfide bonds are basically linking these different chains. Okay, you can see here the first box it's written VL. Okay, in this chain it is written as VH, right? Whereas if you see other boxes, it is C. This is not V, this is C, right? So obviously there is some difference. And what are these boxes called? So this one box is called domain. Okay, so we do not call it a box, we call it as domain. Okay, so you can see there are two domains here, right? Students, clear? So these boxes are
Yes, sir.
domains. Okay, you can see a black region here. This depicts the hinge region. Okay, I hope you understand what is hinge. Have you
Yes, sir.
heard this term hinge?
Yes, sir.
क्या होता है hinge?
Sir, it's a type of joint which allows movement up to a certain
Yes,
degree.
yes, yes, right? So we will also understand what is this hinge, why it is there, does every antibody has hinge, okay? Then we will also understand what is this CH1, Cl, Vl, Vh, right? So we will also understand this. Then you can see this, there are two big chains, right? So these big chains are called the heavy chains, okay? And then there are two small chains which we call as light chain, right? So basically your antibody molecule is consisting of how many heavy chains? Students, how many heavy chains can you see here?
2.
Yes. So one molecule of antibody has two heavy chains and two light chains. Okay. You just need to add one more word here, and that word is identical. Okay. So a molecule of antibody consists of two identical heavy chains and two identical light chain, right? So this and this, they are identical. Okay, clear? Clear to this point? And then you can see below heavy chain, there is some, like, what is this? Alpha, gamma, delta, eta, mu. What is this? Okay. Even below light chain, you can see there is kappa, lambda, right? What is this? Right. So we will understand what is this. Not today. Most probably we'll come to this point uh, in whenever we have the next class. Okay. So at this point, you just understand the concept of domain, right? So these are the different domains. There is a hinge region. There are two identical heavy chains, two identical light chains. Okay. Another very important thing that you need to see here is they have put this arrow, right? So this point, above this point, you have the antigen binding region, right? Which means the antigen will bind here in this region, okay? And rest of the region, it has the biological activity, okay? Students, again, you need to understand, antibody का काम सिर्फ antigen को bind करना नहीं है, okay? So one part is binding to the antigen, right? एंटीबॉडी का एक और काम है टू एलिमिनेट, राइट, टू किल the pathogen, right? The ultimate aim is to kill the pathogen, okay, and remove it from the system, okay? So for that, it need to do certain things, okay, and those certain things are done by the other region, right? So this region is for the biological activity and the above region is for the antigen binding. Okay, clear? Any questions?
No, sir.
Okay.
No, sir.
Okay, good. Okay. So now as you have already seen the structure, right? So you just saw that there were four peptide chains, right? Two identical light chains, right? And two identical heavy chains. Okay? So two identical light chain, two identical heavy chain. So total of four peptide chains, right? And the bonding of heavy and light chain, how were they bonded? They were held together by mainly the disulfide bond, right? You just saw that in the picture also, the disulfide bond. And there are some other interactions like the covalent bond and the non-covalent interactions. But the most significant one is your disulfide bonds, right? And you already just saw this in the pictures. So these are the disulfide bonds which are holding these four chains together. Okay? Now, the first region which you saw was the variable region, right? So the first 110 or so amino acid of amino terminal region. Okay, so okay, let's come back to this picture. Okay, so we are talking about the first 110 amino acids. So if you start from this point, okay, so if you start from this point, this is the amino terminus. So you start from here, right? And till this point. Okay, so there are around 110 amino acids. Okay, so the first 110 amino acids from the amino terminus, they form the variable region, and that's why this is V. Okay, V means variable region. Okay, and why this is VH? Because this is the variable region of the heavy chain. Okay, and this is the variable region of the light chain. Okay, so now you understand what is VL. So students, what is VL? Okay, so what is VL? What is VH? Is it not clear?
So VL is the variable region of the light chain and VH is
Yes.
variable region of the heavy chain.
Yes. And what do you mean by a domain? Okay, so see, this is a peptide chain, right? And it is composed of amino acids, right? So जो पहले 100 या 110 amino acids होंगे, they form the first domain. So all these domains are of around 100 or 110 amino acids. Okay, so one domain consists of around 100 or 110 amino acids. Okay, so there are different domains here. Okay, so we have now understood what is VL, what is VH. Okay, so the first 110 or so amino acid of amino terminal region of both the light or the heavy chain, they are your variable regions. Okay, so now you understand what is this VL and what is VH. Okay, Now this variable region also have some complementary determining regions or the CDRs. Okay, so we will talk about CDRs in the next class. Okay, and these CDRs are present on both the VL and the VH. Okay, CDRs on both VL and VH constitute antigen binding site. Okay, so there are some regions in this variable region where the antigen goes and bind, right? So suppose this is the Y, right? So this is your antibody, right? uh, if you see this first region, suppose the first part of my finger, right? This is one domain. So this domain has specific regions, okay, which actually bind to the antigen. So suppose this is your antigen, right? So this antibody is not going to bind it like this, कि कहीं से भी bind कर लिया, okay? It will bind through its specific regions in the variable domain. It will bind it from those regions like this, okay? So we will talk about these CDRs in the next class. जैसे जैसे things will become clear, okay? So पहले 110 amino acids जो थे, that was the variable region, and rest of the region is the constant region domain. Okay, so rest of that is constant region. Okay, and that is written as Cl and Ch. So students, again, what is Cl and what is Ch? What is Cl? So you can see here Cl, Ch.
So the constant region of the light and heavy chains.
Yes, okay. So the first 110 amino acid, they forms the variable region of the light or the heavy chain, and then you have the constant region domains of the light chain and the heavy chain. Okay, so everyone, is it clear? Okay, so now how this structure was initially deduced. Okay, so there were two scientists, Rodney Potter and Gerald Edelman. Okay, so Potter and Edelman, they were the one who deduced the basic structure of antibody, and they also received the Nobel Prize in 1972 for deducing this structure of antibody. Okay, now these two scientists, they use different methods. Okay, so what Rodney Potter did, he cleaved the antibody with a specific enzyme. Okay, so Potter ने क्या किया? He took the antibody, he cleaved the antibody with the help of some enzymes, okay, and then analyzed the results. Okay, whereas Edelman, he had a different strategy. What he did, he dissociated the antibody by reducing the disulfide bonds. Okay, so now you know antibody के structure में there are a lot of disulfide bonds. Okay, now what happens if you break those bonds? सारी chains अलग हो जाएंगी, right? So Edelman, he dissociate the antibodies by reducing the disulfide bonds. Okay, so इनकी दो अलग अलग strategies थी, we will discuss that. Okay, and because of this work we were able to see or understand the structure of antibody. Okay, so now let's see what Potter did. Okay, so as I said, he used an enzyme. Okay, and he used the enzyme papain. Okay, So obviously ये जो papain है ये एक enzyme है, it will cut the peptides, okay, it will cut the chain, okay. So ये जो papain था, it produced two identical fragments. So you can see here, ये जो papain था इसने कहाँ cut किया, above the disulfide bond, right? Okay, so this enzyme, it made a cut in this chain just above the disulfide bond in the hinge region, okay, and this produced two identical fragments. One of the fragment was your FAB region, okay, basically fragment antigen binding. So I showed you in the very first picture that the initial part is antigen binding, okay. So this region has antigen binding activity, and other was the FC region, this one, okay. So now we just say it's the constant region, right? But at that time when it was discovered, right, it was termed as fragment crystallizable, okay, because it crystallized during cold storage, okay. So उस टाइम तक ये नहीं पता था कि it's a constant region, clear? ये हमें I think next class में पता चलेगा why it is constant region. But when it was initially discovered, it was fragment crystallizable. Okay, clear? यहाँ तक?
Yes, sir.
Yes, sir.
Okay, and why FAB? So AB you can see antigen binding. Okay, so ये था पॉटर का काम. उसी टाइम पे साइंटिस्ट भी था जिसका नाम था अल्फ्रेड निसनॉफ, ओके. He also used an enzyme. He used pepsin, okay. And when the antibody was cleaved with pepsin, it only produced single fragment, okay, having two FAB regions, right? So FC region not recovered because ये जो pepsin enzyme था इसने ये जो बाकी का region था उसको एक मिनट. So FC region was not recovered because pepsin digested into multiple fragments. तो ये जो FC वाला region था इसको बहुत ज़्यादा cleave कर दिया था. So this region was not here. But what was observed was this region, right? And pepsin ने कहाँ cut किया? It made the cut below the disulfide bond, okay. पपाइन ने कहाँ कट किया था? अबव द डाइसल्फाइड बॉन्ड. पेप्सिन ने कहाँ कट किया? बिलो द डाइसल्फाइड बॉन्ड, ओके. ओके, सो स्टूडेंट्स अभी आई एम जस्ट स्टॉपिंग हियर, ओके. सो हमने दो एंजाइम्स देख लिए हैं, राइट. नेक्स्ट क्लास में वी विल टॉक अबाउट the work done by Edelman, okay, and then we will get into the details of antibody, okay. So if you have any questions you can ask. So we have 2-3 minutes for discussion, so now it's your turn. So if you have any questions you can ask. Okay, students, so I will suggest uh, that you read about the structure of antibody. Okay, जितना at least मैंने पढ़ाया है, so that in the next class I will obviously ask all these things and then we will move ahead. Okay, so just try to read this, and if you have any question you can ask now, or maybe when we start with the next class. So if you have any question, we can discuss them at that point. Okay. Okay, students. So I think I'll end the class now. Okay. So see you in the next class. I hope you have understood what have been taught today.
Yes, sir.
Yes, sir.
Okay. Okay.
Thank you,
Thank
sir.
you, sir.
Yeah, I'm ending the
Thank
class now. Okay.